Hi everyone, in this talk I will be presenting the anatomo-functional organization of hyperdirect pathway to the subthalamic area in human connectome project subjects and Parkinsonian patients. Subthalamic nucleus is one of the classical deep brain stimulation targets in Parkinson's disease, and it involves in different pathways such as direct, indirect, and hyperdirect pathways. And we have already now some three-parted subdivisions regarding the direct and indirect pathways without having any clear-cut boundaries. We also know that the anteromedial axis of SCN-DBS we would have some psychiatric symptoms, and that's why we also investigated another surgical target which is located on the anteromedial part of the STN that we call medial subthalamic region, which has been using as target for non-motor symptoms. We wanted to see if we could have functional differences between these two regions, and we wanted to obtain subject-specific sub-parcellisation of this region regarding the hyperdirect connectivity, which would be helpful for a better targeting in DBS surgery. We used two different datasets, 30 healthy subjects from Human Connectome Project and 20 Parkinsonian patients. All diffusion data were multi-shell. All data were pre-processed, then we used multi-shell, multi-tissue constrained spherical deconvolution framework, and we generated 100 million streamlines using probabilistic tractography, and then we filtered them to the 10 million streamlines by using spherical informed filtering techniques. All anatomical segmentations were done with T1 weighted images. By using our segmentations and our tractogram, we can extract our streamlines of interest. For example, here you see streamlines between Brodmann area 6 and STN. We can do same for MSR. But one of the important points is the anatomical priors. In order to isolate only the hyperdirect pathway, we excluded striatum pallidum and cerebral peduncles, and also we added cross-exclusion between two structures. Here is the first results. These are the streamlines of STN and MSR. The green streamlines are coming from sensor motor cortex, blue streamlines are coming from associative cortex, and red streamlines are coming from limbic cortex. We are able to see differences between these two structures in the individual level, and here are the average number of streamlines for both datasets. Darker bars are for STN and lighter bars are for MSR. As you can see, STN has higher connectivity with sensor motor areas, whereas MSR has higher connectivity with limbic areas. From here, we can go toward the individual segmentation by using tract density maps, which will provide us cortical connectivity profile for each voxel on our regions, and then we can group together voxels which have more similar profiles and create our clusters. Once we have our clusters, we need to check their profile in order to understand the functions. For example, here you see on the x-axis Brodmann areas and on the y-axis tract density values. We can add much more colors regarding the function of the cortical areas. For example, this first turquoise cluster has higher connectivity with sensor motor cortex, more specifically with Brodmann area 4 and 6. We can do same interpretation for the other clusters. Second cluster has mixed connectivity, and third cluster has predominantly limbic connectivity. We also obtain same kind of cluster profiles in PD dataset. These results were also reproducible across multiple subjects in two datasets, and our message was the anatomo-functional gradient that we observed from more lateral sensor motor part toward the more medial limbic part. As a future perspective, we are also acquiring age-matched control dataset to quantify the alteration in Parkinson's disease. We also have perioperative clinical data that would provide us some anatomical clinical validation of diffusion dataset results. Thank you for listening.